Hi, I'm Jenna Fi, and this is Amalia Arcio Hoskin, and our project is about the effect of microgravity on the reproduction of Curli-producing E. coli 0157H7 strain 43895OR.
E. coli 105787 is a single-celled organism known as a bacteria. This bacteria is gram-negative and rod-shaped. It is pathogenous, which means a disease-causing. It is a cause of foodborne illness. Contamination is a fecal-oral. This you can get it by eating contaminated meat or crops or swimming in contaminated water. If E. coli is contaminated, it can lead to bloody diarrhea and can lead to a kidney failure. E. coli 105787 strain 43895OR, mutant strain, which was discovered by the USDA after preserved simply. It was originally obtained from the CDC of E. coli 105787 was thawed and prepared for analysis. Mutant exhibit Curli, which are external features that contribute to the pathogenicity of bacteria by helping them produce biofilm. Mutation is not always exhibited, and it can be manifested in one generation, but not again in another.
The strain and biofilm. Biofilm are the result of cells sticking to a surface and then to each other. Biofilm have been shown to increase a species' pathogenicity or disease-causing. Species readily formed biofilm are highly disease-causing. In 2006, USDA studied biofilm formation on glass, Teflon, and steel, and compared the strain's biofilm growth capabilities with a non-Curli forming strain of E. coli. As you can see, OR was greater, which means it produces hardy biofilm.
Our experiment. What effect does microgravity have on the reproduction of E. coli 105787 and on the external structure known as Curli? As the space travel continues and with private companies looking to play a role in space, it is important to know what effect microgravity will have on the pathogenicity.
We used type 3 for our experiment. Our main volume was 6 milliliters of tryptic soy broth. For our short ampule A, we used 0.9 milliliters of approximately 1 million colony-forming unit of the strain. For short ampule B, we used 0.92 milliliters of glutaraldehyde as a fixative. To start the experiment, we crack short ampule A and shake, which activates the experiment. After 6 days, we crack short ampule B and shake, which deactivates the experiment. Due to the bacteria's disease causing, we were not able to handle the samples ourselves. So we worked with Dr. Harold Alvey of Indiana University Northwest. We worked with USDA to get the strain and do the experiment. We used Skype to join Dr. Alvey in his lab. We walked him through the steps of our experiment.
Real science. On June 1st, we were told that there was a miscommunication and our experiment was deactivated early. We were awaiting a reflight opportunity in this coming fall, and we'll be analyzing our ground truth and flight samples from this current spring and summer trip in the reflight.
We will be using a scanning electron microscope, SEM, to analyze the outside of the bacteria to determine if Curli are still present and what effect, if any, microgravity had on the structures.
Team. Amanda Arcio from the community director, Nicole Gustafson, our teacher, and my name. And this was a great opportunity, and we want to thank you, the Indiana Space Grant Consortium, the NCESS, and the NanoRacks, the USDA, especially Dr. Gail Ulican, and Dr. Harold Alvey in the Indiana University Northwest. Thank you. Any questions?
All right, thank you for an excellent presentation. If you have any questions to this team of researchers, please come to the microphone.
What led you to do this experiment?
Excuse me?
What made you do this experiment?
Oh, well, we were really interested in bacteria because the year before we did paramecium, so we wanted to do something different. So we decided to do E. coli because it was dangerous and disease-causing, and we wanted to try it and take the risk.
Will it lead to any further studies?
Not that we know of.
Okay.
Thank you.
All right, thank you again.